I'm Neriman Yilmaz, and together with Dr. Marcus Wilken, we are welcoming you to the first FABI International Seminar Series of 2023. Soon, there might be some light switch due to the load shedding, so don't panic, we are here, everything will be under control. But it is also my pleasure today to introduce our speaker, Dr. João Araújo. And he's currently the curator mycologist of the curator of mycology at the New York Botanical Garden, and his research interests are the insect-associated fungi and their associated mycoparasites. So he combines the fundamental taxonomic science with natural history, field work, evolutionary biology, microscopy, and the amazing photography that you will see soon. So today, Joe will take us to a journey that combines the world of science, nature, and entertainment, and we'll talk about to us Ophiocordyceps, zombie ants, and The Last of Us. And Joe, thank you again for accepting our invitation, and we are really very keenly looking forward to hear your presentation.
All right, thank you very much, Neriman, for such a nice introduction. So today I will be talking about the biology behind the zombie ant fungi, which has gained a lot of attention the last months, I would say, because of the series, The Last of Us. And it has been a boom, so everybody talking about cordyceps, talking about fungi that turn insects into zombies and all that. So just to mention how mycology has been pictured in movies or in games. So there are some very famous movies and games. For example, the Alice in Wonderland has been showing lots of mushroom-oriented stories. And the Mario Bros that I used to play since the late 80s, that this video game has been around. It also heavily relies on mushroom on the main topic on the story. But if we notice, if we pay attention, almost every time that fungi are included in the screen, in the movies or games, there are mushroom. The story is always around mushrooms and Amanita more specifically. So very few other fungi have been gained attention in the screens. So that happened recently with The Last of Us, as we have mentioned, which is a series that created a story that is about the cordyceps, that is a fungus that actually exists, actually Ophiocordyceps. I will be talking about that later. And that they have switched from infecting and manipulating and zombifying insects to become a human pathogen, which have been decimating most of the human beings on Earth. And 10 years ago, it was first released as a game. And more recently, this has been released as a TV series that has been really success, and everybody talks about that. So, but I'm here to tell you that the real-life zombies are much more interesting than what has been exhibited in these shows, in these video games. So the real life is much more interesting. So the kingdom fungi is comprised by about 135,000 species and estimations ranging from 600,000 to almost up to 10 million species yet to be described. And most of the knowledge we have on mycology is about decomposers and mushroom-forming species and mycorrhizal associations because of their economic and ecological importance. Also the lichens that are bioindicators of the air quality and good model systems to study symbiosis. And of course the plant pathogen as has been played a really important role to study and investigating these fungi more in depth because they are economically important and also ecologically important. But we know very little about the fungi that are associated with the most specious group of animals, which are the insects, which is quite surprisingly because these insects are everywhere. So we would tend to think that the fungi associated with these insects are everywhere. It would be relatively better known, but it is not. It's really a dark side of mycology, I would say. Here I will show you an overall view of the main lineages of entomopathogens across the different phylum. So in basidiomycetes, which is one of the most diverse group of fungi, we have only two lineages associated with insects as parasites. So one is a Fibler rhizoctonia, the termite, a fungus that mimic the termite eggs. And on the right side there's Septobasidium, which is the Septobasidiales, the order that actually they make these scale insects their own slaves. So they don't kill the scale insect, but they penetrate hyphae of the scale insects just to reach the sap from the plant and to get nutrition out of that. So the insect's only the gateway into the plant, apparently. And in the case of the egg mimicking termites, so these orange spheres are fungosclerotia. And when these termites go, they are blind. So when they go out to forage and they feel these spheres mimic the termite egg in their shape and even the scent they release. So they are really easily mistaken by a termite egg, and they bring these fungosclerotia into their nest. And eventually this fungi can switch to the parasitic stage and then eventually consume all the eggs and really collapse the colony. Another case is the Blastocladiomycota, the so-called chytrids. So they also infect insects. There are lineages infecting insects. So in this case here, it's interesting life cycle because they can infect two different microorganisms, the copepods and the mosquito larvae. So when they infect the copepod and then they fill the whole body of this organism, and then when it's completely filled with zoospores, it will break, and then the zoospores will swim in the water, in contact with the mosquito larvae and penetrate through its cuticle. And then, as you can see here on the right side, all these cells are inside the mosquito larvae. But it can either kill the mosquito larvae and then go to the next stage, to the copepod, and then cycling back again, or it can remain inside the mosquito larvae until it reaches the adult stage. The adult stage, after the first blood meal, it will lay a fungal sporangium filled with zoospores, and that ensures that the fungal spores will be laid in these breeding sites of the mosquitoes. That ensures that there will be suitable hosts around it. So that's really a brilliant strategy, I think. And then we a more speciose group of entomopathogens, which is the Entomopteromycota or Mycotina, depending on the author, they consider as a phylum or not. And so we have a large diversity of entomopathogens here, and they also display the behavior manipulation, as we can see here in this beetle, taking the edge of the leaf and making this display, the mating type display that is caused by as the fungus grow inside the insect, it will spread their wings, and that mimics the mating display. So when other beetles and to try to mate with this infected beetle, it will eventually get in contact with the spores and get infected. Yes, as we can see, it's a really interesting biology, and that's very different compared to the cordyceps. To start with, these fungi are really difficult to culture, so that poses a challenge to work on, especially in genome scale, because we cannot culture it most of the times. So another example within this group is the Massospora that many of you might have heard about, which is in fact the periodical cicada. Some species only occur every 17 years following the cicada life cycle. And what it does, it's one of the very few lineages that can sporulate from the living body of the host. So as the cicada still acts relatively normally and can even fly, it will spread spores everywhere it goes. So as you can see here, it's missing basically half of the abdomen, but it is still apparently acting normally. And when you try to mate, it will eventually infect other cicadas as well. So that's really an interesting group. And now we're going to narrow to the ascomycetes, more specifically the Hippocrealian fungi, which cordyceps and Ophiocordyceps belong to. and that has the group that has the largest number of entomopathogenic species. So to give you a background on the evolution of these species, so let's step back in the Cretaceous around 150 million years ago, and then we will have the radiation of the angiosperms. So all these land plants were gaining territory and then speciating and then creating all these new niches to be exploited by other organisms, including the insects. So the insects, as the angiosperms were radiating, the insects were following all this radiation and these new opportunities of niche to be explored and developing different mouth parts to adapt to different types of plants or different parts of the plants like fruits or flowers or pollen, nectar, or sucking the sap out of the plant, like parasitizing the plant. So there are a myriad of different lifestyles of the insects that were co-evolving with these already evolving angiosperms. And together with that, we would expect that pathogens and parasites would follow this trend of evolving and speciating. So if we trace like this green box is the Cretaceous. So here we're comparing the plants, insects, and entomopathogenic fungi phylogeny, and we see that in the Cretaceous there are a lot of events of splitting here. So that means that new lineages, from one lineage now we had two lineages, and that's how the speciation occur. So, and then we see that speciation in plants followed with the speciation in insects and the entomopathogenic fungi. So it is parsimonious to hypothesize that plants evolving and insects co-evolving with them, and entomopathogenic fungi were following all these evolutionary chains that were occurring back then. So that's was a three-layer of biodiversity. And as the name already mentioned, so the zombie ants are ants that are manipulated by these Ophiocordyceps fungi, and they are really interesting and good for the fungi, and really a great resource to be explored because they are everywhere in the forest. They're super abundant. They comprise a huge amount of biomass in the forest, so it is a really great resource to be explored because not they're only like large in biomass, but there are lots of different species, often close related species, living together in the same type of forest, in the same location. Sometimes even have some niche overlap, and that promotes the fungus, in this case the parasites, to jump from one close related species of ant to another. So then the speciation is more likely to happen because these ant species were promoting the fungal biodiversity because they are excellent resources to be explored by these parasites. And there are also great model systems to study parasitism and evolution and many other types of perspectives you wanted to study, because ants are really well known. So they are ecologically dominant, so they are really common in forests in everywhere. And their phylogenetic relationships is well known, so it's easier to build cophylogenies with ants and fungi and to understand when these speciation events occurred, because we understand the host really well. And we know their behavior, their life history, their rich fossil records, which help us to calibrate phylogenies and then understand when certain events happened. And they are most abundant animals in tropical forests and has a huge biomass, as I mentioned. And one very interesting and important feature here is that the host remains identifiable after the fungal infections, because in many cases, for example, the Coleoptera parasites that infect beetle grubs in the soil, so they're soft-bodied insects. So often the fungus destroy the taxonomically important features that we use to identify that host. And so it's more complicated to identify because we have to do often use a forensic kit to extract really tiny amounts of remaining DNA of the host. So that makes things more complicated, more expensive. But the ants and other hard-bodied insects like wasps or even moths, they're relatively the features are relatively conserved. So for the ants it's the same, because they even after months in the forest, after fungal infections, it is possible to identify the hosts based on their morphology. So that makes this system really, really amazing. All these things I listed. And also the identification of the host is feasible and it's doable, even still in the field by specialists, of course. But that makes this system really interesting to study that. So here I will step back then and come back to the question that after The Last of Us people often ask me, which is cordyceps can infect humans. And then I have to explain that cordyceps are ophiocordyceps, because there's a misconception, and even myself, I often see myself referring to cordyceps as the ones that manipulate. That actually the ones that has the manipulation abilities are ophiocordyceps, not cordyceps. So until 2007, all these cordyceps-like fungi, they were included in a large family, Clavicipitaceae, and they were considered as superficially as cordyceps. So everything that has a clava, and in fact the insects, the ascomycete, was in this batch of species so-called cordyceps. But then the Sung and Joe Spataphora and others, so they did a comprehensive phylogeny, a really hugely cited study in Studies in Mycology in 2007, where they proposed a new arrangement, a new taxonomic arrangement for the cordyceps. That was a groundbreaking study that really paved the way for everything we do in cordyceps systematics and taxonomy, really based on this important study. So this yellow ring we can see here around the phylogeny is the Cordyceps sensulatu. So that includes families within Ophiocordycipitaceae, Clavicipitaceae, and Cordycipitaceae, of course. But this figure shows that the genus Cordyceps, the sensu stricto Cordyceps, are those species. Here there are more, but represented in this phylogeny is this part of the tree. So that's the Cordyceps sensu stricto, the genus Cordyceps. And in blue here, we have the genus Ophiocordyceps. In red, we have the zombie ant fungi, which are the manipulators, the zombifying lineages. So we see that cordyceps and ophiocordyceps, and especially the zombie ant groups, like this is Ophiocordyceps unilateralis, and this here is Ophiocordyceps bucketii, that also do the behavior manipulation. And we have, as you can see, there's multiple origins. There are also one or two lineages here that are not presented, but the origins of behavior manipulation are multiple. So they independently evolved. So, but that's something we're looking to that now, actually. So, but this slide is to show that cordyceps is one thing, Ophiocordyceps is another thing, and the zombie ant fungi, the Ophiocordyceps unilateralis and others, are within the Ophiocordyceps genus. And that they cannot infect humans. Oh, here I had a close-up. Because as this study, we show that there is a really one species of fungus for one species of ant. So the specificity is really high. So one species within the Ophiocordyceps unilateralis will only infect that species, particular species of ant. It cannot infect the sister species. So that it makes it really clear that these fungi are super strict regarding the host they infect. So if they cannot infect even its closest relative of that ant species, imagine jumping from ant to humans, mammals that has the physiology and the immune system completely different from the ants. So that's really unlikely that these fungi will jump to humans, at least in the next million years from now. So it's really unlikely. And now we're into the biology and showing the major features of the zombie ant fungi per se. And here we have the stereotype phenotype of a zombie ant. So we have an ant infected by fungi. We see these brown hyphae often emerging from the joints of the ant and the typical biting behavior. And in tropical forests, it takes about, according to laboratory experiments and what we see in the forest, it might take around 2 weeks to develop the fruiting body, which has this stalk, the cinema, that has asexual structures, the Ixutella type, and it has this beautiful ascoma. And if we make a section of this ascoma, you will see all these beautifully arranged perithecia that will actively shoot spores when it is mature. So we have preliminary data showing that one a single ascoma like that can shoot more than 300,000 spores. So that's really, really dense. Imagine a tree that has a dozen of these fungi, mature fungi, shooting spore, so times 300,000. So imagine around that tree how many spores it will be. So it will be literally a minefield. So other ants that belong to that same species of the host that pass by these spores, we will eventually get in contact with the spores and get infected as well. So this is another 2-minute video showing a species that coincidentally I described in exactly this place in Rio Claro in Colombia back in 2018. We published in Studies in Mycology, and Netflix, they went there and did a time lapse, beautiful, the most beautiful time lapse on zombie ants that I ever seen. And I'm happy to share that with you. So this really, really amazing video. I think I watched that over 100 times, but every time it's like if I was watching that for the first time. So I think this is really beautiful. And this slide shows a bit of what it is to be doing field work, which to me is one of the most important parts, because without collecting the specimens, we don't have anything to study. And also being there is really crucial to have insights on their biology and how they shoot spore, how they reach out to the next host, how they transmit, what their strategies, behavior manipulation. In order to understand all that, it is impossible without going yourself to the field and immersing yourself there. So here's a slide showing some of the places I have visited. And these fungi are really tricky to work because they get moldy, and many other opportunistic fungi are on top of them, of course, because they are in the forest, so many other spores land on top of these fungi. So if we keep them in a tube for maybe 2 days, and sometimes even the next day, there will be already other fungi, very often verticillium, growing on top of these cadavers and on top of the entomopathogen. So we often need to work on the improviser lab, makeshift labs. For example, this bottom left here was in a boat in Rio Negro in the Amazon, and on the top right is in a hotel room in Ghana, and here in the bottom right is when I was infested by a mite or a poison ivy. I never figured what it was, but it was really, really difficult. And here is we're doing field work, myself and Charissa that is here watching the talk. So really good times collecting fungi, maybe around 10 years ago or so. So that was good time. So that field work is really amazing. And next I will just show you a bit of how it is to be in the forest looking for these fungi. This one I recorded in December when I was in the southern Brazil in a cloud forest. So there's really like Lord of the Rings type of forest, I think. It looks like an orc will jump from behind these trees on us. So I think this is really, really a photogenic and really interesting forest. And all these epiphytes, mosses and ferns and all these plants usually are microclimates that these zombified ants seek to bite. So there's a species that specifically bite this type of moss. As you saw in the Netflix, they always were biting into a moss, a bryophyte. So that they're not just specific in the host they infect, but in the place that the fungus will lead the ant to bite is also species specific. It's very niched. I will show in the next slide. So that's how we usually find them, like biting these hanging epiphytes, mosses, and ferns. And that's a bit of the process of describing species. So we collect them in the forest, taking photos, and back in the lab we do sections to access their morphology, their morphological features. We do DNA sequencing, and then we deposit them and make it publicly available. And I have an improvised for now. We are looking to do a more professional site within the NYBG server. But for now, I have my own Flickr account where I have all my photos. I would be happy to share the link if you are interested. And as I mentioned, processing these fungi properly is really crucial. So here we see a beautiful cinema of Purpuricillium, which is beautifully purple, I think. But if you don't process the specimens, as I said, other fungi eventually can grow. So if you store these Purpuricillium in a bag without silica, without drying it, look how it's actually beautiful, the community of many other sorts of fungi that grow on top of them. So that's why we need to process these fungi really quickly. Otherwise, other fungi will grow and that specimen will eventually get lost. And this shows a bit of what is the foundations of all the work, which is taxonomy. So describing species, going to the field, describing species, and sequencing them and putting them in this phylogenetic framework is absolutely the foundational work that is needed in order to understand evolutionary patterns and ecological patterns and behavior manipulation, evolution, and many other aspects. So back in 2014, 15, so of all these fungi of Cordyceps unilateralis, which is nowadays is comprised by, if I'm not wrong, 32 species. So it was represented in the phylogeny as just one branch of Cordyceps unilateralis. But I did my PhD, and also other researchers, especially Jennifer Luangsaard in Thailand, has done a brilliant work to describe these species in Thailand. It has been contributing a lot to entomopathogens in general, but in these zombie ants as well. So now from one branch in the tree, now we have over 30. So we really amplified this, we open up this single branch that was before, and now we have a much better understanding either in the Ophiocordyceps unilateralis clade and Ophiocordyceps niphophioides clade that also do the behavior manipulation, and the one that was the second evolutionary origin of behavior manipulation, which is down here, the Hymenosteuboides, that has the hymenostube and type of anamorph, which is the secondary origin. So we studied these two different groups, and we could describe species, new species, doing behavior manipulation with different strategies as well. And I often get asked how many species of zombie ants there are. So here is just a preliminary data to try to understand how is the potential, how large is the potential for new discoveries, and how many species will be within this group. So we did really intensive field work in Brazilian Amazon, in Reserva Duque, near Manaus, Maracá, and Viruá. And we also had a colleague that is worked with ants there for over 20 years. And he has really a deep understanding on the Amazonian ants. So those are genera of ants, which is represented by this gray bar here with number of species. For example, Pheidole has 72 species in the Amazon, and Camponotus has 32. Now I think it's 34, but let's stick with this, 32 here. So it's really fascinating that Pheidole is completely free of infection, while half of the Camponotus species in the Amazon are infected by Ophiocordyceps fungi. So what makes Camponotus really magnets for fungal infections, while Pheidole that actually inhabit the same environments? We see Pheidole and Azteca ants everywhere in the forest, but we never found them infected. And we found Camponotus everywhere. Pachychondria is also common, Dolichodorus a bit less, but Camponotus is really, we found dozens every single day we go out in the forest. So that's one question that intrigues me, why some ants or insects are free of infections while others are magnets and promote fungal diversity. So this I'm showing the ant, but this pattern also repeats in insect orders. For example, Camponotus here could be Hymenoptera, so there's lots of species infected. Coleoptera, Lepidoptera, and Hemiptera, which are the most specials, they are really common to be found. But other insect groups are completely free of fungal infections. So that's really an open question yet. So if we extrapolate like that half of the Camponotus species are infected as they are in the Amazon, this is conservative. We know with preliminary data that there are more, but let's keep it official. So 50% are infected by 50% of the Amazonian Camponotus are infected by Ophiocordyceps fungi. So if we extrapolate there to the worldwide number, which is around 1,200 species, and half of those, conservatively, half of those will be infected, so we are talking about 600 species of these fungi to be described. So we described around 30. So there's still 570 species yet to be described, just belonging to this clade, Ophiocordyceps unilateralis. So there's a huge diversity yet to be discovered. And every time I go to these forests, I find new species. Even here in the United States, we're describing one from Pennsylvania. We found biting onto a rock, which is, we never saw it before. But this hidden diversity of complex species is not only restricted to the zombie fungi or the Ophiocordyceps unilateralis. I will have another slide to show that in more detail, show the morphology. But now I will go around this Hippocralian tree just to show some examples. So mycoparasites associated with Ophiocordyceps is a complex. Gibellula is absolutely understudied and has a huge diversity. Acanthamyces on adult moths, or Hypocrella ascarsonia type of fungi on scale insects and whiteflies. Basal lineages in Ophiocordyceps infecting beetle larvae. And these ones on cicadas, which that's one lineage that I'm really focusing right now. So now I will just show some photos of these complexes that I collected in my last trip to Brazil in December. So here we have a scaled insect, this bump in this twig, covered by this white hyphae with this beautiful orange, yellow perithecia superficially growing on them. So we didn't sequence that yet, so we don't know which fungus this is. So, but I believe this should be a Cordyceps type of fungus. Maybe I'm not risky here, but we'll see. So let's wait the results for the sequencing. We'll work on that. So we have also the Orthoptera. So this cricket we found in Brazil. It's quite large cricket infected by this unidentified fungi. It resembles a bit Purpureocilium, although it's not purple, but we need to study that further. We just collected. This is a Nigelia, which infecting this beetle. It's a really beautiful specimen. We found maybe 4 or 5 of those in that trip, which this is another specimen. I believe it's the same species, just grew in a different way, more into the asexual stage. But we can also, if we look more closely here on the right side, we see this fruiting body. These tiny dots are perithecia. They're starting to grow there. And this is one of my favorite species now, which is still undescribed. And I don't even know which genus is this, because we haven't sequenced that yet. But I believe will be a Cordyceps as well. Probably closely related to the Gibellula / Torubiella type of thing. But we still need to sequence those. It is in Brazil right now. So another photo of that. So I think this is really beautiful. We found 5 of those in a single tree attached by a fungal hyphae. If you see the legs are attached in the substrate by extra sticky fungal growth. There's another perspective of that. And the Purpureocilium, that is a new species close related to Purpureocilium atypicum. And we found that in Brazil. And this could be a new species that it already got a lot of media coverage. The Guardian covered that discovery, and maybe more Scientific American will release a note on that. And that's a new species of Ophiocordyceps we found in the southern Brazil, also biting bryophyte. And this Hypocrella, Morleriella type of fungi infecting these whiteflies. And here on the left side, we even have Conoidocrella. So here we have at least two entomopathogenic species basically on a single leaf here. So we have 2. So it's the underside of a leaf can be a whole universe when we find these associations. So that's really strike me every time I go to the field. And really strange looking caterpillars, the hairy caterpillars, has these still immature Ophiocordyceps growing. Ophiocordyceps dipterigena, that even the head of this fly fell, but you see these 2 beautiful fruiting bodies coming out. And that's the Cordyceps orbovaria we have to sequence to make sure, infecting a hairy caterpillar as well. That's a close-up of it. And now we'll be talking a bit about the behavior manipulation aspects of that. So we are running out of time. So we published 4 years ago hypothesis from in which circumstances the behavior manipulation occurred, so evolved, how it originated. So our hypothesis is that it occurred because of the niche overlap between beetle a fungus infecting beetle larvae in the forest floor. Here we see a number 1 in this photo here in the forest floor. And here we have number 2, which are the zombie ants, where they occupy nowadays. But here we reconstructed the phylogeny of Ophiocordyceps, and we did ancestral character reconstruction coded according to the host association. So here what matters is that red is the ants and blue are Coleoptera, beetles. So our results reconstructed the common ancestor of the genus Ophiocordyceps. It was a lineage infecting beetle grubs in the forest floor, in these trunks, exactly as these basal lineages of cordyceps still do. So if you go to the forest and find beetle grubs infected, that's likely that it will belong to Ophiocordyceps in these basal clades here. For example, Ophiocordyceps melolonta is a complex of species, and they are often found in these logs in the forest. So the transition occurred here in the node number 2. So from a beetle, as you can see in blue, so one lineage remained as a beetle associated, but other lineage jumped from beetle to the ants, which is in red here, is shown in red. So as you can see, they jumped here, and all these zombie ants and behavior manipulation and all this biology apparently promoted biodiversity, because from a single jump we can see an event of speciation and lots of new species being described for this clade after this transition. This can be also sample biased, because I was working a lot to describe these new species, and so far as I know there's not an effort dedicated, for example, to describe species infected to Coleoptera, for example. So, but in my opinion, there is an effect here, and there is a effect that this behavior manipulation here plays a role to in this speciation. So back then, the ancestral infecting beetle grub would have jumped from this beetle grub here, solitary, completely naive, and there's no defense, and then they jump to the ants. So, but if we think about the ants, they have a feature, they exhibit a feature called social immunity, which is the public health system of the ants, let's say that. So this collective behavior helps the nest to be clean. So when they see one of the nest mates infected by a pathogen, a parasite or anything, so it displays some symptoms of disease, these ants will be ripped apart, killed, and their remains will be thrown out from the nest or put in these trash chambers and isolated from the rest of the colony, because they don't want a disease to be spread in the colony. So according to our paper, so our hypothesis that the social immunity was the driving force for the development of the behavior manipulation, because once this lineage here jumped from the naive and solitary soft beetle here that was defensiveless, and when they jump to the ants, the ant colonies that also inhabit these trunks still nowadays, they're carpenter ants. So they carve their nests in these logs. So there's a niche overlap. So when this fungus jumped from the beetle to the ants, they found this pressure of social immunity, and eventually one lineage evolved the ability to go under the radar for a while, so the others cannot recognize that as a sick ant. And before the nestmates recognize it and then activate the social immunity against this ant, the fungus remove that ant there to place it in the summit position, a higher position, when it will shower spores on the forest floor, creating these minefields. And when the ants go out to forage, they get infected by these spores. But the fungus is then avoiding the social immunity and then able to complete their life cycle, which otherwise it wouldn't be possible, because the ants would destroy the host and consequently the fungus. So that's the theory that's accepted now about the driving force that caused the behavior manipulation evolution. And there are different types of behavior manipulation. So they're not just the summiting, that species get infected in the soil and climb upwards. So in this case, which is one of my favorite species, so they nest in the canopy, but when they are infected, they descend, and then to die in this base of the tree, they're covered on moss. The ones we found are invariably Octoblepharu albidum, so it's a specific species of moss. And then the ant is manipulated to bite there to dig a little bit and bite to the base of this moss. However, when the fruiting body of the fungus comes out, so it looks like that, and the sporophyte of the moss looks really, really similar to the fungus. The fruiting body of the fungus resembles really closely to this sporophyte of the moss, and the fungus drives the ant to die on that moss. So there is a behavior manipulation to protect that, and also the morphology of the fungus mimics the morphology of the moss. So there are many different types of match there. So they match morphology, so they match the behavior of the ant with that moss. So I'm really convinced that's a really interesting story waiting to be told here. So I'm looking forward to explore this system further. This is, so far as we know, specific to the Amazonia region in South America. And here's another type of behavior manipulation caused by Ophiocordyceps nymphophioides. So when the ants are infected, they try to find these crevices or these cracks or these places where they can hide a bit and bite onto this wood, or sometimes there is moss also. But the anamorph here, they grow in a really interesting way. So remember that ants has the social immunity. So when other passing by ants find these corpses here, they will take it and throw them on the ground, so to avoid that tree and consequently the way up to their nest to have a fungus inoculum there, so that transmitting disease there. So they try to clean that up. However, look at these structures here. So these structures, they grow out of this ant, but they look that they're growing on the tree. So what they do, they grow from here, from the thorax, and they go downwards, and then they cripple underneath the bark. And eventually they pop up these structures, as we can see a bunch here, or these type of structures that produce spores at the tip, conidia, asexual spores. And then even if the other ants remove this cadaver here, and look how many we find at the base of the trees, like many dozens of cadavers that were removed. But in that tree, the ant cadaver will be removed, the fungus inoculum, the fungal spore-producing structures will still be there because they are anchored in the wood, in the trunk, and just the host was removed. So the ants are not smart enough to clean up the fungi. Well, they would get infected anyways. But anyway, so that's really a morphological strategy to overcome the social immunity. So there are different types of strategies to win this arms race between fungi and the ants. Here I show the number, and this is showing how different it is, the growth from tropical forests compared to temperate forests. So here in the tropics, in the the Amazon of Brazil, so just 3 days they grow this stalk, while in the temperate forest in South Carolina, they take a whole year to complete their life cycle, because they get infected here in the summer, and then when the fall comes, it gets colder, and then this fungus stop, and then winter it got literally frozen, and then we just resume its growth in the next year when the summer comes again. And if you notice, they're really specific in their microclimate. So if they bite onto leaves in the temperate forest, the leaves would fall on the ground and the fungus would not be able to develop. So the lineages in this temperate forest, they adapted, so they don't bite the leaves, they bite onto twigs. So we were curious about that, and what we did was to do an ancestral character reconstruction here, and we coded according to the biting substrate. So green means biting leaves, and brown and blue means biting twig. So we found that the ancestral was biting leaves in the tropics, most likely, and as they were gaining more latitudinal gradients towards temperate forests, they had to adapt to these different environments in different climates. And we found that these evolved at least 4 independent times. And if we plot these records in a map, we see a clear pattern that green means biting twigs, leaves, and brown means biting twigs. So in the tropics here, it's really clear that there is a trend of biting leaves, and in the temperate zones here, bite onto trunks, with 2 exceptions, which is this specimen. I found just one guy biting trunk in Ghana, and this guy here that bite leaves in the cold weather in Japan, but they bite onto evergreen plants. So they're evergreen forests that never lose their leaves, so it doesn't really matter. So this is an adaptation to the colder climates. And now just to wrap up, so a bit more of a different types of behavior manipulation in the moss, and here some species drive their host to bite specifically into palm tree spines, which is pointed downwards, and every single morning there will be a drop of dew there, ensuring that the fungus will have a supply of water. So I think this is a really, really beautiful manipulation that they hang in these spines. So it's quite interesting to see that in the forest. So I'm skipping that because of time. And now I'll talk about, as a last topic, about the mycoparasite, which is a work I have been doing with my colleague Charissa at the University of Utrecht in the Netherlands. And we have found just from one species of zombie ant fungus, Ophiocordyceps campanoti floridani, we collected in Florida. So we found different types of mycoparasites. So this had a perithecia sexual state, and this was the asexual state, this cinemata here producing these very interesting phyalides. And at first I thought that this would be the teleomorph and this would be the anamorph state. But when we sequenced that, we found out there were two new lineages within Cordyceptaceae and within Ophiocordyceptaceae. So they actually belong to two different families. And when we sequence that and look at the morphology, they look very different from anything I have seen before. And the phylogeny shows a really unique branch for these two lineages. So we named these two lineages a new genus, so Niveomyces and Rubielomyces. So there soon will be more species being described for Niveomyces from Asia. So it is being reviewed right now. So this study was published in Personia in December, so a few months ago. And many more will come for sure. And just to finish, so zombie ants are really great tools to communicate mycology, communicate science, biodiversity, parasitism, evolution, and also helps to reach out the broader public, because with these series and documentaries and these time lapses that National Geographic and BBC and many other media channels are producing help a lot to spread the word about mycology and how interesting these fungi are. Actually, I got interested in zombie ants and cordyceps because I watched that BBC movie over 13 years ago, and that blew my mind. So here I am 13 years later studying cordyceps because of that. And also the arts that also has been explored, especially in the past, for artists. Actually, this one here I even have in my own hand. So it's Shimizu, Daisuke Shimizu. He has a really amazing book, yeah, which is this. So he described their species and also made really beautiful illustrations of that. So this is a really beautiful book. So thanks a lot. Thank you all for staying here with us, and thank you Neriman and everybody at FABI that invited me, and it was a pleasure to be here, and I hope next time I can be there in person. Thank you very much, you all.
Joe, I wish you were here and you can hear us like, wow, was it real? Like, what was it? Like, it was, I think, really mind-blowing. I must say, I'm so jealous. I mean, those things definitely look so beautiful, but I know how much it takes to take photo of them. It's not like you have a great camera and the fungus so you can take the photo. It is definitely your talent is speaking there as well. So the floor is open to the questions from audience. Please feel free to ask your questions. But just to break the ice, I will ask one question, because you mentioned that how it like development of these basically the structures takes, for example, in Amazon forest it was a different time, and in US it was a different time. So for those time-lapse photos and the images, how long did they take on average? Do you have an idea?
Yeah,
I
well,
don't think it was a year, yeah.
yeah, the last one, I don't know if you noticed, but it didn't work out because these fungi are really sensitive. So that time lapse I did with a National Geographic photographer 2014, like almost 10 years ago. And we just removed the leaf because we just found that ant biting that exact moment. We were very lucky. So we waited, it bite the leaf, and we removed just 10 meters away just to set up the camera and all this equipment. And then the fungus didn't grow as we expected, just because we moved it like 10 meters away and we detached the leaf, it was biting. So they're really, really sensitive. So just another information. And if we remove it from that original location, probably it won't grow as it should. So the hyphae will grow all unorganized, so it won't form this organized stalk. Anyways, so it's really sensitive. But in order to do that, that footage, the beautiful one that Netflix did, was in Colombia. So that's a tropical species that I was not there. I was invited to be there, but I was doing the biodiverse course in the Westerdijk. So that I was really dreaming about doing that course. So I said no to that documentary to do that course, which I don't regret. But my colleague Tatiana San Juan, she was involved in the supporting them. But I believe that time lapse was set for about 2 weeks or so.
Yeah, manageable, I think. Regarding the method of infecting certain genera and not others, he is asking. I'm noticing maybe it's okay, Jit, don't worry. I'm noticing maybe a discrimination on body size, many small ants in the non-infected, and possible critical morphology, smooth versus structures too. Is there any insight in possible correlations there?
Yeah, well, I don't think size it's super important here. Can I share my screen again just to show a
Yeah,
photo?
sure, sure.
So this paper here we published, let me show the smallest species ever described. So it's millimeters. So this guy here. So this ant here is like 3 millimeters. And the fruiting body is so small that it can only fit in the same plane 6 perithecia. So it's really tiny. So it's very tiny. So I think if the size would be crucial, these species wouldn't be infected. And other species that, like, for example, some ponerians that are very large, they are rarely infected. So I don't think the size plays a huge role on that. So my ideas, my hypothesis on what's going on, I think it should be related to the microbiome of these ants. So perhaps they have endosymbiont stage of the Ophiocordyceps that is blocking the parasitic stages to infect them. So I don't know if you have heard that, but there's some lineages of Ophiocordyceps that switched from parasitic to become endosymbionts, especially in hemipteran insects. This has been known for I don't know, 15 years or 20 years, but it was never described as proper species. So we're working on that as well in Japan. So yes, there's these lineages of Ophiocordyceps that can be parasites, and there are cryptic lineages that are beneficial endosymbionts. So I think that maybe these endosymbionts could work also as a bodyguards against the pathogenic lineages. Well, this is just hypothesis. I've never tested anything, but I think that this would relate to the microbiome of these ants. Or, for example, the Camponotus, they have a gland. They don't have a gland there that other ants has. I forgot the name of this gland. But there's something in Camponotus that only Camponotus doesn't have. So this can hint that this is also a feature that can probably make Camponotus more susceptible to diseases. But my educated guess, since I never tested any hypothesis yet, is that the microbiome that these apparently uninfected ants might have some answers. That's my hope.
Okay. And we have another question in the chat box, and Hari is saying that, wow, wonderful. Are there any chances of Ophiocordyceps human infection in an event trauma or immunocompromised states, or a prolonged exposure as the basically saying that can Ophiocordyceps affect the people when immunocompromise happen? Because I'm sure you've seen that thing on Twitter that plant pathologist apparently got infected by the fungus that he was working with. So do you think in that case that can happen?
Yeah, I have read not the whole article, but I have read something like that recently, maybe 2 weeks ago,
Yeah.
that a guy that worked with fungal pathogens got infected. Yeah, so far as I know, there's no record of Ophiocordyceps infecting even immunocompromised. But I never read the study, but I have seen around information that there's a few cases of immunocompromised patients being infected by Bovaria. So I know that there are medical cases on that. Well, but if you think about that, there is a schizophilum community infecting immunocompromised patients as well. So probably immunocompromised people are susceptible to any microorganisms eventually. So I wouldn't be surprised if a super weak person, immunologically speaking, would be susceptible to any fungal infections. But so far as I know, there's no record of Ophiocordyceps or even cordyceps infecting the humans, so far as I know.
I saw Mike's hand is up, but I want to give Yin his question. Yin is asking that if we want to culture this Ophiocordyceps fungi, any tricks that can make it happen in the lab?
Yeah. Well, we started doing that a long time ago, especially Charissa that is here. So she is the master of culturing these fungi. We have done that together many times. And we found that the best stage to establish culture is the early stage of the fungus. So just right after the fungus has make the biting, so the next few days will be the ideal. So when we still, up to the point we still see the stalk, the pinky stalk just starting to grow, that's the good stage to crack open the abdomen, and so that endosclerosion, that plug, so we basically remove it and put on agar. So if you bury it on agar, we'll have problems to grow. So you have to just put them superficially there, and this really slow growing fungi. So in order to reach like these size colony, like millimeters, takes months, because these fungi are not meant to grow a lot. So their universe of growth is the ant body. So they are designed to not to grow like a mushroom that will spread the hyphae on the ground, but they're to be more condensed in the ants. But there's another way to establish cultures, which is shooting ascospores. But I have tried that a few times, and the single ascospores, when they are shot and they are not in contact, they are spread in the slide or in the agar plate. So we will just grow secondary growth. It's called Capliconidia-4, bearing a Capliconidia, like we saw in that video, the fly that has secondary growth. So Ophiocordyceps also has this secondary growth, which is actually a taxonomically important feature. But if we shoot a bunch of spores and they clump together, like making a mount of spores, I have seen ascospores establishing cultures in this way. But single ascospores in the zombie ants, I never seen them germinating and forming like colonies, unless they are clumped together, like hundreds of spores. Or the early stages, when you find the ant with early stages, you dissect and you crack open the hyphal bodies and you just plate it. So those are the two ways to culture them. But they are really slow growing. You have to be patient.
Well, firstly, thank you for a really beautiful talk and just an amazing subject. I was interested in a comment you made. You were estimating the numbers of your cordyceps, how many there might be in the world. And you suggested that maybe, and I don't know why you did that, but you said sort of 50, let's say 50% of these ants have Ophiocordyceps. Why 50%? Why wouldn't you say that every species of ant has a species of Ophiocordyceps? Was that, I mean, it was just interesting.
Yeah, thank you. Thank you for the question. Yeah, well, 50% was the rate we found in Brazilian Amazon. I'm sorry if that was not clear. So those 3 spots I showed, we did more intensive surveys. And we worked with a myrmecologist there, and he knew all the species that were there. They do these inventories. So we found that half of the Camponotus species were infected by its own species of Ophiocordyceps. That was part of my PhD. We found that half were infected. So if we extrapolate half of the Camponotus we found infected in Brazilian Amazon to the global number of Camponotus species, which is around 1,200 species, if we keep that same, as I said, conservative rate of 50% are infected, we would expect that half would be infected, so 600. But as I said, it's conservative. I know I have data showing that it's more than 50%, because every time you go to the forest, we find one species of Camponotus that has not been recorded yet. So I think it's just a matter of searching, and perhaps, which I don't doubt, that each species of Camponotus would have its own species of Ophiocordyceps. I wouldn't be surprised. Does it clarify a bit?
Yeah, no, that's very nice. Be interesting to see what happens in the future. Thank you.
All right, thank you.
And then also we have one question about again culturing from João, and she's asking that I ever work for Cordyceps militaris infected on one of the larvae, I cannot pronounce. Problems when it's mass production, very difficult to get the fruiting body. Would you share the good media to produce the biological
Right. Well, as I don't know if it was in the talk. I think Neriman and we were talking about sequencing and establishing cultures. So because it was complicated for me as a student to work on all this paperwork to export live cultures from so many countries we visited, so I optimized the protocol to sequence DNA out of the specimen. So although we established cultures, especially for the North American species, we haven't been establishing cultures for all these species yet. So my next project will, because we're doing phylogenomics, so we will need that. But I don't have a large experience in the culturing Cordyceps militaris, but I have many friends that has been dedicating their lives to do that, either commercially or for scientific purposes. So I will just put my email here in the chat. So please let me know if you want to. I can put you in contact with some people. I have a colleague, he's one of the leader experts, Nigel Jones. So he works in a pharmaceutical company in China. I have spent some time with him there, and they produce cordyceps in really large trays. So it's really beautiful. But I don't know what they use. And coincidentally, recently I have revised a paper that tested the cordycepin content across different types of media, and they found that particular groups of insects will promote the production of cordycepin compared to other insects and even compared to agar. So I would recommend you to perhaps try to find this type of studies, or you can get in contact with me through this email, and I can direct you to some colleagues doing really great work with culturing, especially Cordyceps militaris.
And by chairing this session, I take the advantage of asking the last question as well. But I see on your maps you show Africa only from Ghana. So I'm asking to you and Charissa, when are you coming so that we can do the collection of those amazing basically Ophiocordyceps species in Africa?
Yeah, I would love to go because Africa is really a black box for entomopathogens. So there's studies in the Amazon, there's some studies that Jennifer do in Thailand. So there's some really traditional work made in Japan by Kobayashi and others, Maines in US. So there's here and there, but Africa, there's absolutely nothing so far as I know. Some studies from Madagascar and some studies from the 70s that Harry and Rob Sampson did a brilliant foundational work in the 70s there in Ghana. And I have been to Ethiopia as well, and I found amazing specimens there, but because of other problems, we couldn't work on them. And yeah, so it's really amazing that Africa has so many interesting lineages, and I'm looking forward to visit Africa again. And that would be really great if we could collaborate in a project on cordyceps and their mycoparasites, and perhaps establish cultures, deposit with you guys there, which I have deposit cultures with the ambrosia beetles from Florida. Yeah, so that would be amazing. Let's talk about that and plan that for sure.
I see thumbs up from Charissa as well, so that will definitely be the plan. Therefore, I'm handing it over to Bernard to make the final comments on that, and also I think that also closure for the meeting as well.
Yeah, I think everybody here would say, well, we're waiting for you, hopefully soon. You've blown our minds, you've infected us, I think. If we weren't infected already, it's very, very hard to not be infected after such a beautiful talk and such fascinating work. Wish you all the best with this continued discovery and amazing our biological, illuminating this biological diversity, and we're waiting for you. We're waiting for you.
Yeah, thank you very much. Thanks a lot. That will be a pleasure.
And thank you so much for the time and the effort into the talk, and everybody that's joined, and Neriman and Marcus for setting this up. We really appreciate it.
All right. Thank you very much. Please
Thank
feel
you
free to
again.
reach out if you have questions.
Thank you, everyone, and see you next time.
Obrigado, Bárbara, em português.
Bye. Obrigado.
Bye.
Bye.